Welcome to a Taconic T-Onco webinar entitled Preclinical Mouse Models to Address Vaccine Efficacy in Oncology, given by Dr. Jean-François Mirjolet from our French oncology partner company, Oncodesign. Jean-François has been working for Oncodesign for 9 years as a study director and the head of cellular pharmacology. Since his arrival at Oncodesign, he has played a central role in the development of reconstituted human immune system mouse models, mice. Jean-François obtained his PhD from the University of Nancy, France, in cellular pharmacology of anti-cancer drugs, followed by a postdoctoral position at the IRIBHM in Brussels, Belgium, in chemokines and chemokine receptor implication in cancer cell migration. He is now the technology director at Oncodesign, directly supporting business development activity and working closely with clients to select the most appropriate study designs for the evaluation of their therapies through preclinical studies. We hope you enjoyed today's webinar.
So the topic of today is preclinical development of cancer vaccine. Before enter into the subjects, I will give you a bit of history. The father of vaccine therapy is a French doctor who names Louis Pasteur, and it is the first having performed a preclinical experiment in chicken in 1880, and he is also the first having explained the principle of vaccination. He also is the doctor who has cured the first human who has been bitten by a rabid dog. The main advantages of a cancer vaccine over standard therapies is that this therapy should also be a targeted therapy, meaning that there is some specific antigen expressed by tumor. This treatment should also be a long-lived efficacy, having recurrent expression and activation of T lymphocytes, and this vaccine therapy should also be safe, as side effects are only expected as local reaction with very, very minimal systemic toxicity. The main component of a vaccine should be and has to be the selection of the right antigen in order to specifically target the tumor cells and in order to avoid autoimmunity, which is the main issue with the vaccine therapy. The cancer vaccine should also contain a right adjuvant in order to activate in a potent and robust manner the immune system, meaning antigen-presenting cells and related effectors. And at the end, the effector that is activated by the vaccine should be responsible of an innate and adaptive response with the preferential induction of effector of choice, meaning T cells, cytotoxic T cells, and not regular T cells. For treatment purpose, therapeutic vaccine in oncology has to be therapeutics and not prophylactics. However, knowledge on vaccination was first acquired on infectious disease, and some also tumors are related to infectious disease, such as HCV for hepatocarcinoma, EBV for some lymphoma. However, in that case, many tumors are not related to vaccine, to virus-induced pathology, and then we have to find specific targets and specific antigen other than viral proteins. The expected mechanism of action of vaccine therapies is first capture of tumor antigen. Second, processing and maturation of this antigen by dendritic cells. And at the end, generation of the effector population, meaning cellular response by T cells, but also humoral response by B cells. Actually, there is a various platforms in clinical trials, and the various platforms are related to the type of vaccine. There is 4 to 5 different types of vaccine. The first is composed of antigen and specific antigen for tumor cells, and this antigen could be peptide, proteins, or even antibody. The second platform in clinical trials is recombinant vectors mediated vaccine, where the antigen of interest is expressed by viruses, yeast, or bacteria. Some platforms also use whole tumor cells as immunogen, and in that case, there is a possibility of autologous or allogenic injections of tumor cells. And at the end, the last platform in clinics is a cellular therapy, meaning directly injection to patients of antigen-presenting cells. Regarding the potential targets for therapeutic cancer vaccine, there are a lot of potential targets of interest. Among others, there are oncoproteins, which are most of the time mutated or overexpressed protein in cancer. As example, there is mutated RAS or p53, and as overexpressed or modified expressed protein, there are R2 or abnormally glycosylated MUC1. There is also as potential target the oncofetal antigen that is normally not expressed in normal cells, but re-expressed in case of cancer. In this type of targets, there is CIA, glycopeptide, and also NY-ESO1 antigen. There is also target associated with abnormal differentiation of tissues like PSA or GP100. As I said before, there is a virus-related tumor cancer, and in that case, target of interest could be a viral protein like E6 or E7 viral protein of HPV, and recently identified there is target of interest regarding stem cells treatment like OCT4 or TET protein. I would like to explain a little bit the mechanism of action of all these various platforms, and the end efficacy is for sure for all platforms the activation of a specific immune response. However, for each platform, the whole process is a little bit different. So starting with antigen vaccine, the first is identifying a peptide or protein specific for defined type of cancer. This protein could be either naturally produced or laboratory generated. The main issue for this type of antigen-based vaccine is that the treatment will be either cancer-specific or could also be very general, and also there is a specific case for lymphoma with anti-idiotype vaccine. The next platform is a recombinant vector-based vaccine, and in that case, there is no direct injection of the antigen to patients. What is injected is a vector that will express in situ the tumor antigen. In that case, the objective is to associate a non-host protein that will induce a high immune response, and to associate with this non-host protein the tumor antigen, and in that case we will increase and specifically induce the immune response toward the tumor antigen. The next case is the tumor cell vaccine, and in that case the antigen is a global protein extract coming directly from whole tumor cells, and it is better to induce apoptosis death as compared to necrotic death. Apoptotic cells will generate, in many cases, a better and specific immune response. This tumor cells vaccine could be either autologous, meaning the tumor cells are coming from the patient and injected after a preparation, or could also be allogenic, and then the tumor cells are coming from another patient and prepared before being injected for a therapeutic purpose. The next mechanism of action is the cell-based vaccine. In that case, we used the target is to prepare antigen-presented cells in order to avoid the first steps of the immune response, meaning the capture of antigen and the processing of the antigen by APCs. And in that sense, the APCs are collected from the patient, derived ex vivo, meaning that they are loaded or transformed to express and present the antigen at their surface ex vivo before being re-injected to the patient in order to generate and activate a specific immune response. The challenge with the cancer vaccine is that we need to obtain a high-level immune response. The tumor-associated antigen could be specific for patient or specific for defined tumor sites. So it could be an issue to generate many specific vaccines for only few patient treatments. There is also an issue regarding the antigen that could be differentially expressed between the primary tumor and the secondary metastatic disease, meaning that the vaccine will cure the primary tumor but not affect the metastatic growth. And at the end, the main issue is that the tumor microenvironment is probably the worst environment to mount an immune response, as the tumor microenvironment express many regulators of the immune response. I will now give you a few examples of the preclinical evaluation of the vaccine and what is the prerequisite to perform preclinical evaluation of this vaccine. First, we need a model where we have an intact immune system. For sure, the vaccine will activate the immune system, and we need many actors of this immune system, meaning DCs for antigen-presenting cells, as well as effector of the immune system, T cells, B cells, NK cells. For efficacy purpose, we need also to have a tumor target in order to not only monitor the immunomodulation of the immune system, but also to analyze the anti-tumor related effect, and in order to also target antigen in the microenvironment in order to improve efficacy of cure. we need a complete tumor microenvironment, meaning fibroblasts, immune cells, and extracellular matrix components. At the end, we also need assays to analyze the immune response, meaning to analyze if we obtain specific immune response. And the best model for doing that, for sure, is Syngeneic model, as in this model we have an intact immune system and also the ability to engraft Syngeneic tumor. So I will start with this Syngeneic model. In collaboration with Taconic, so for the T-Onco portfolio, we offer a large set of Syngeneic model, meaning that we have spontaneous and experimental metastasis Syngeneic model, like the 4T1 breast carcinoma. We also have various settings of tumor cells engraftment, like subcutaneous model, but also orthotopic bladder in rats and mice. And this is a huge choice to improve new vaccine therapies in this type of model. So I will show you 3 examples of the literature explaining how to analyze vaccination therapies in this Syngeneic background. The first example is related to whole DNA vaccination in order to test the best way to induce response to DNA vaccine. So the protocol is immunization with R2 DNA-coated gold particles. The model of interest is D2F2 mouse mammary tumor that were modified to express human R2 antigen. And at the end, there is many readout of efficacy of the vaccine, meaning analysis of specific response through ELISpot assays or pentamer staining, or analysis of the specificity of the humoral response. So in this paper, the author has tested 2 different ways to inject this DNA into mice, either Jengen injection or jet injector. And there is 2 groups of treatments, a group where they inject R2 expressing DNA and a mock vector. What they have demonstrated is that by pentamer analysis, meaning detection of very specific T cells, there is a huge increase of the number of specific T cells only in the group receiving the R2 targeting DNA and injected through Jengen, and nothing was observed in the jet injector groups, either in the periphery or in the spleen of these animals. The second readout was the quantification of specific IgG, and again, the author confirmed that only in the Jengen deliver DNA vaccine, they observed R2 specific IgG that are of IgG1 type, but also of IgG2A and 2B type, and nothing was induced in the jet injector groups. So, as a conclusion for this first example, this model was a suitable model to demonstrate differences in terms of efficacy between gene gun and dead injector delivery of DNA vaccine. And in the case of gene gun delivery, this DNA vaccine produce a very strong Th1 polarized response with specific CTL expression and also specific secretion of antibody. The second example is based on the protein therapeutic vaccine, protein for VEGF, targeting VEGF, and the model, I selected the model where they use a CT26 mouse model. So the process of this study is many rounds of immunization, tumor challenge, and follow-up of the efficacy of the vaccine as anti-tumor response, but also specificity of this vaccine. 4 groups were tested: placebo, the antigen alone, the adjuvant alone, and the complete vaccine, combining both antigen and adjuvant. So what they have found is for first the anti-tumor efficacy, there is a very marked decrease of the number of metastases only in the complete vaccine, meaning antigen and adjuvant. This is evidenced by the total number of metastases, but also by the size of the metastases, meaning the total metastasis area. Regarding the immune response, the titer of specific antibody was tested is 2 different strains of mice, the C57BL6 mice, but also the BALP-C mice, which is the strain for the CT26 model, and they have found that there is significant differences in terms of the intensity of response, meaning that the immune response was far better in the BALP-C background as compared to the C57BL6 for both the total antibody titer, but also in terms of quality of this antibody, meaning that the antibody produced by the BALP-C strain is more efficient in terms of inhibitory function as compared to the one produced in the C57 BALP6 background. So the main finding of this study is that using a protein-based vaccine, they obtain a specific cellular response, and the efficacy was only not only related to primary tumor growth, but also evidence in the secondary disease, number and size of metastases. What also is interesting in that case is that probably a model and the strain of choice for this vaccine therapy is BALP-C mice as compared to the C57BL6, where the quality and intensity of humoral response was lower as compared to BALP-C mice. The last example is regarding a complex peptide-based vaccine. The target in that case is aberrantly glycosylated MUC1, and the model used was a model transgenic mice expressing human MUC1. The process was of treatment was immunization intrademally 3 times before tumor challenge with subcutaneous injection of tumor. So the author have tested various combinations of this complex peptide. The first is the structure combining a TLR2 agonist, a T-helper epitope, and obviously the MEK1 epitope. Four other groups were also treated with part of this complex peptide, meaning only TLR2 and T-helper structure, only T-helper MEK1 epitope, or only TLR2 ligands. So what they have found is, first, regarding anti-tumor efficacy, is only with the complete structure, meaning the three parts of the peptide, they obtain a very, very nice efficacy as compared to the untreated groups regarding the tumor size, and we observed a decrease of efficacy when only part of the peptide are used separately. Again, they have compared the ability and the efficacy of the immune response. So they have prepared serum from immunized animals, and they have performed cytotoxicity evaluation of this antibody, and they have found that lysis is far better when using serum from mice immunized with the complete peptides as compared to the other groups. And again, they have analyzed the specificity of the cytotoxic T cells response, and these assays also confirm that in the group treated with the complete and complex peptides, they obtain a far better release of interferon gamma, meaning that the number of CTL produced is higher in this group. So, as a conclusion for this example, it is and it could be very interesting to generate complex peptides as antigen for initiating a specific and potent anti-tumor immune response. That it is also interesting to note that the covalent linkage of the three components is essential to generate maximum response. If you cut and co-inject the three parts of the peptide, you will obtain a response, but lower than the covalent peptide. And they also have demonstrated that the CTL produced in that case are able to recognize both normally and abnormally glycosylated MEK1 expressing tumor. So, as a conclusion for this Syngeneic model, this is a model of interest to evaluate vaccine, as these mice have an intact immune system. They are easy to handle, and a large choice of models are available, meaning injection of tumor IP, IV, subcutaneously, even orthotopically. And the analysis is very rapid, meaning 3 to 4 weeks in order to obtain the first results in terms of immunomodulation. However, the main issue with this model is that, for sure, mice are not human, and the mutation profile of tumor in mice is specific and of species, meaning there is differences between the tumor in mice and the tumor in human, and the aggressiveness of this model could also be an issue. There is also, for supporting the needs of having more human model, two main publications describing that there is lots of difference in terms of immunology between mice and human. First, the structure of the immune system. There is lots of lymphocytes for mice, and most of the leukocytes there are neutrophils in human. In terms of effectors, they are not the same in mice and human, meaning the antibody classes are different. There is no CD89 in mice, and this Fc alpha receptor are expressed in human. And also the cellular arm of the immune system is also different between mouse and human. Recently released publication show also that the genomic response in case of inflammatory disease are not the same between mouse and human, and that mouse model sometimes are poorly mimicking the human situation. I have only selected the third figure of this publication, showing that the pathway activated in human for human burn, human trauma, or human endotoxemia situation are not the same in human and in mouse. The intensity of activation is absolutely not the same, and sometimes there is opposite effect, meaning that for the repressed pathway, we have some that are repressed in human and some that are induced in mice. So, in addition to Syngeneic model, there is a huge need to develop model where we have a humanized immune system. So, what kind of model are available? There is two main possibilities. The first is using PBMCs, meaning functional human immune cells, but in that case there is a graft-versus disease issue. Some function of the immune system was already evidenced, meaning this model for vaccine purpose could be suitable. So recall response is effective in this model. We can observe immunomodulation, but, and I will explain why later, related anti-tumor efficacy are still to be demonstrated. The other option for humanized mouse model is model based on stem cells. In that case, there is still huge characterization to perform, meaning functionality for all cells, all immune cells are to be demonstrated. However, this model was already tested for infectious disease-related vaccine and show potential interest. However, this model was not yet used for therapeutic cancer vaccine. Again, in the T-Onco portfolio, the model we have available is using PBMCs in various mouse strains, or also using stem cells in adult or newborn nog animals. I have also chosen two different examples from the literature showing the use of this humanized mouse model for evaluation of antibody, for therapeutic cancer vaccine. The first model used BALB/c-RAC2 knockout IL-2 receptor gamma knockout mice, so the BRG strain, and this strain was humanized with intrahepatic injection of stem cells coming from human fetal liver. After establishment of human immune system, the mice receive intramuscular injection of already available vaccine, meaning vaccine against HPV or tetanus vaccine. What authors have demonstrated is, yes, this model could be suitable for analysis of immune response related to vaccine treatment. They demonstrate that half of the immunized animals generate specific IgG in response to HPV treatment, and also they have compared between responder and non-responder the number of the concentration of IgM and the concentration of IgG, showing that in this type of model, this type of vaccine could generate a specific humoral response. They also have analyzed the cellular response, meaning the number of B and T cells between the vaccinated group as compared to the control group, and there is a specific increase in the vaccinated group, and they also demonstrated that between responder and non-responder there is a huge difference in terms of B cells and T cells absolute number. So the main conclusion for this study is this humanized mouse model is suitable for vaccine study, and they observed B cells expressing specific IgM. This IgM repertoire is the same as the one that should be expected in human. An issue is regarding the number of responders, which is only 40% in that case. However, we know that in human settings, there is also a huge heterogeneity in terms of response regarding vaccination. Some humans are good responders, meaning after the first vaccine injection, the immune response is already effective, and in some cases, after 2 to 3 injections of vaccine, the appropriate immune response could not be generated. So the heterogeneity observed in that type of study could also be an interest to identify biomarker of response if non-responder and responder animals are analyzed in detail in order to understand the differences between both populations. The other example is using NHG mice, but in that case engrafted with human PBMCs. There is 2 different settings. The first is a long protocol, 9 weeks with injection of a small amount of PBMCs. The vaccine is autologous DC vaccine, meaning that monocyte-derived dendritic cells was generated ex vivo and pulsed with MART1 antigen. And the second protocol is a short protocol where 10-fold higher amount of PBMCs was injected, and with a shorter delay between injection of PBMCs and vaccination using prepared dendritic cells. What they have found is, in case of there is no vaccination, obviously no specific T cells could be identified through MART1 multimer analysis, and only in case of the short protocol, this specific T cell population, specific to MART1 antigen, was identified in these mice. Regarding the efficacy of this identified CTL, this cytotoxic T cells was specific and effective in terms of tumor target lysis. So, as a conclusion, using PBMCs and grafted mice, vaccine therapy could be evaluated in a short protocol in order to avoid graft-versus disease, and then analyze the immune response before the occurrence of graft-versus disease. The author have demonstrated that in that case, the antigen used and presented by ex vivo prepared dendritic cells are suitable to generate a specific response, and so humanized mouse model based on PBMCs could also be a model of choice to evaluate efficacy of therapeutic cancer vaccine. So, as a conclusion for this humanized mouse model, the strengths are: we have a human immune system, so we are not faced to the mouse differences between immunity, differences in terms of immunity between mouse and human. The number and the number of models, the choice of model is also huge in that case. We know that we have many models related to human disease as IPIV, subcutaneous or autotopic human, and for sure we are closer to the human pathology. However, there is some issues with this humanized mouse model. They are hard to set up. As you have probably understand, the identification of the protocol from the previous study, we've seen that the short protocol only is suitable to use PBMCs, and right now the all-in-one, meaning human immune system and human tumor, are not yet available. So we have the ability in humanized mouse model to observe the immunomodulation, meaning the reaction and response of the human immune system. However, we are not able right now to analyze the anti-tumor efficacy related to this immunomodulation. So, as a general conclusion for the vaccine evaluation, as you probably all know, the first therapeutic vaccine was approved by the FDA in 2010, and it's called Provenge and it is a cellular-based vaccine and approved for non-symptomatic metastatic prostate cancer. At least two others are close to approval, and they produced positive results in clinical phase 3. One is based on GP100 peptide vaccine for locally advanced melanoma, and the other is anti-idiotype vaccine, so a protein-based vaccine for follicular lymphoma. As a conclusion regarding preclinical evaluation of vaccine, sure, non-complete and perfect model exists. There is interest to understand and to have an all-in-one model to perform studies in mouse, so using the Syngeneic mouse model. However, as I've shown, there is a lot of differences and sure regarding immunity between human and mouse. So it is interesting to develop and set up humanized model. The perspective regarding the evolution of humanized mouse model is to try to set up a model where we have the ability to have a complete match system, meaning HLA-A2 expressing mice, HLA-A2 positive immune cells, but also HLA tumor. We are going to develop and set up this type of model in collaboration with CIEA in Japan, and we have obtained a grant from Jose Onedo for that field. We have other developments regarding improvement of humanized mouse model, but for other applications. An application is IL-2 transgenic animals for ADCC-mediated efficacy of antibody, and we have also developments regarding humanization of the mouse liver. I will now thank you for your attention, and I will answer all the questions you may have.
Thank you, Jean-Francois. That was a great seminar. For people who do have questions, please go ahead and type those in the Q&A panel. We have received a couple. The first question would be, what about GEM cancer models for preclinical evaluation of vaccines? Is that something that you can do?
Yes, sure. The genetically modified mouse is also a model of choice to evaluate cancer vaccine. The main issue with this model is the establishment of the model, but when you have it, it is a model of choice as we could obtain a cancer pathology that is closest to the human, meaning the tumor is naturally occurring in this model. There is no inflammation generated through the injection of the tumor cells or the engraftment of the tumor fragment, either in syngeneic or xenogenic situation. So this is an advantage of GEM model. The other advantage is that the aggressiveness of the model. We have a GEM model that is long and where we have few weeks, even months to analyze the tumor development. So the window for treatment is longer as compared to the regular syngeneic background.
Okay, our next question is about hematopoietic stem cells. So you talked a little bit about PBMCs, humanized PBMC models for vaccine studies. Are the HSC humanized models, are they suitable for vaccine study, and what would some limitations of those models be?
So I've shown an example for where the author have used a stem cell-based model. So yes, this model could be of interest. However, as I said, only for evaluation of immune response. So the first part of the questions, meaning, yes, we need to generate a specific immune response, but we need also to observe an anti-tumor efficacy related to this modification of the immune system. The stem cells model are suitable for the first part, and there is some publication showing that right now. there is also new improvement, meaning HLA expression of this transgenic model will help to improve and rationalize the use of this stem cell-based model. One missing part is still having a tumor-bearing and humanized animals in order to have a complete picture of the immune response and the anti-tumor response.
Okay, that actually leads right into our next question. So has there been success growing human tumors in humanized mice?
We have succeeded in generation of tumor in humanized background. So we are able to observe growth of tumor in humanized animals, either in PBMCs bearing animals, but also in stem cells and grafted animals. We have used this model for therapeutic purpose, and what we have observed is, yes, we can analyze this immune response, meaning we have generation of specific antibody, we have generation of specific T cells identified through pentamer FACS analysis. However, as I said before, we did not achieve to set up a model where the anti-tumor efficacy is evident, meaning that we have half of the answer, the immunomodulation and the specification of the immune response, but the anti-tumor efficacy are still missing in this type of model.
Okay, we've got a quick question. The CT26 cell line that you showed some work on, is that a colon or a lung tumor model?
It's a colon model, Syngeneic for biopsy mice.
Well, it looks like that's all the questions I had. So I want to thank everyone for attending the seminar. We will be sending out a link for the slides. And if you have any further questions, please don't hesitate to get in touch with us. Thank you all very much.